So, it's my pleasure to introduce tonight's speaker, Jo Handelsman. Jo is the Associate Director for Science at the White House Office of Science and Technology Policy. She is on leave from her appointments as Howard Hughes Medical Institute Professor and Frederick Phineas Rose Professor in the Department of Molecular, Cellular, and Developmental Biology at Yale University. Before joining the Yale faculty, she was chair of the Department of Bacteriology and professor in plant pathology at the University of Wisconsin-Madison. Jo is an expert in communication among bacteria that associate with soil, plants, and insects. She is a pioneer in the field of metagenomics, particularly in the agricultural and medical sciences. She also has done important and notable research on science education and on women and minorities in science. In this capacity, she co-founded the Wisconsin Program for Scientific Teaching, the Yale Center for Scientific Teaching, and the National Academy's Summer Institute on Undergraduate Education. And she co-chaired the working group that developed a PCAST report on meeting the nation's STEM needs in the coming decade. Among many awards and honors, Jo served as president of the American Society for Microbiology and received the Presidential Award for Excellence in Science Mentoring. She earned a BS at Cornell and a PhD in molecular biology at the University of Wisconsin-Madison. Her talk tonight is entitled Microbiomes and Microbiomics, How Microbes Rule Our Microbial World. And please join me in welcoming her to the podium.
Well, thank you, Larry, and thank you all for being here and being willing to think about science and microbiology late on a Friday night. Does the sound okay? Everyone can hear? Okay, great. It's really a pleasure to be here. I've heard a lot about the society since I moved to Washington, and I never thought that I would be elected a member, so thank you very, very much. Very prestigious and illustrious and historical society, so I'm really honored to be a member, so thank you all. Today I'm going to talk about the microbiome, and it's actually a plural, microbiomes, because there are many different ones on Earth. And I thought I would talk specifically about the image of microbes in the world at large, and how our view of microorganisms has evolved toward what is now known as the microbiome era. And I will keep the part on the microbiome itself short and then open it up for questions. So hopefully I can talk about the aspects of the microbiome that perhaps you've heard about or are particularly interested in instead of the ones that I'm interested in. But I'd like to take the historical view first of why we have the feelings we do, most people, toward microorganisms, which are mostly negative. And so to lead up to that, I'd like to start with what microorganisms are, just so that everybody has a sense of what the definition is, and then look at the historical view of microbes as pathogens, and then how that view is beginning to evolve and why. So microorganisms are all of the organisms that we can't see individually with the naked eye. In an aggregate, we often can see them. For example, on a petri dish where we have many billions of cells, or in a fungus, for example, that forms macroscopic structures. So the microorganisms that we know of as the most common and the most interesting for tonight's talk are, first of all, the viruses, which are not really organisms because they can't replicate or make copies of themselves on their own. They depend upon a host cell to do that for them. These are the eukaryotic microbes, and this is a paramecium, the organism that with which I fell in love when I was 12 and brought me into microbiology. And then this is a fungus, and both of those are eukaryotes, which means their cells have nuclei, and that distinguishes them from the prokaryotic organisms that we see here. And these are typical pathogens, but they are representative of the large world of bacteria. I'll mention another group of microorganisms, the archaea, in a few minutes, but these are the old microbes that we've known about for more than a century. So I think that this slide depicts a lot of the origins of our hostility toward microorganisms. When people think about microbes, they think of epidemics. They think of plagues that you hear about in the Bible. They think of Camus' plague, the Black Plague that swept through Europe and killed off a vast number of the people in medieval Europe. And those have been canonized. Those are our canonical stories of the damage that microbes can do. Rarely, if ever, do we see the microbes celebrated instead, because it's much less dramatic than these exciting events where a big portion of the human race is wiped out. That's much more exciting. Another example is the protist, it's called Phytophthora, that caused the Irish potato famine. So this is an example of the eukaryotic type of microbe. And this organism infected the potatoes all over Ireland in the mid-1840s. And due to a combination of just bad luck and lousy weather, and some practices that nobody knew were contributing to the vulnerability of the potato crop, this organism wiped out the entire potato crop of Ireland. I mean, extensively and completely. And the result was, because the British were taking all of the other agricultural products away in rent and taxes, the Irish people had nothing left to live on. And so this resulted in at least 1 million deaths, and then 1 million more emigrations from Ireland to Canada and the United States. So again, if you ask almost any Irish person or person of Irish origin, they will know the Irish potato famine story, and they may even know the name Phytophthora. That's the only time I've found really complicated and largely unfamiliar names become familiar to people, is through that story. Phytophthora, I mean, who learns about that in school, right? But people of Irish heritage have learned this story, because invariably those in this country have origins that they're related to the Irish potato famine, and their ancestors came here because of it. So these negative epidemics, these dramatic, catastrophic events in history are what punctuate our historical view of microbes. But despite this, there were some people all along the way that recognized the benefit of microbes. And that is illustrated here. Does anybody recognize the handwriting? Anyone guess? English? Well, this is the handwriting of Mark Twain. And Mark Twain wrote an absolutely astonishing novel, which was never published, called My 3,000 Years with the Microbes. And the section that I'll read to you from the first few pages goes like this. We know that the human race was saved from destruction in the beginning by the microbe. We know that the most valuable citizen on the earth was the microbe, and that the human race could no more do without him, that's typo, sorry, it should be her, than it could do without sun and air. So here we have Mark Twain in the early 20th century. We think this is around 1907, 1909, pretty close to the time he died, touting the power of the microbes, the positive power of microbes, and saying that they are as important as the sun and air to human existence. This novel is absolutely hilarious, in my opinion, because he writes it from the microbe's standpoint and talks about living in this community of microorganisms and gives this historical, almost epic view of the microorganisms. So his creativity is enormous. But you might wonder, where did he learn about the power of microbes? Because it certainly wasn't the common view in those days. Most people thought of microbes as germs. And it turns out that he was in Connecticut, and there was a professor named Professor Conn, spelled C-O-N-N. And no relation between Conn and Connecticut, even though they're spelled the same way, and he was in Connecticut. We think that Professor Conn was the first bacteriologist in the United States on an East Coast campus. And he was a fabulous communicator, and apparently gave talks, public talks like this one, about microorganisms. And because Twain lived nearby, he came to some of these talks. And in fact, there are mentions, even on the very first page, you can see it says he was studying microbiology under Professor Conn, and he cites some of what he learned from him. So I think it's a really exquisite example of how the positive power of microbes has been canonized because of a public lecture about science. And so what you are doing here in your series, I think has potential to change history and change the future. It's just one, of course, of many examples. So if Mark Twain knew in the beginning of the 20th century that microbes were absolutely essential to life on Earth, why, for 100 years, has this view of microbes as germs persisted? I taught microbiology and I taught non-science majors for years, and I would always ask them at the beginning of the course, what are bacteria or what are microbes? And all I would ever hear is, they're germs, they make you sick. Occasionally, maybe someone would say, oh, they make yogurt. But that would be the only example that they would know of a benefit of microbes. And that has just begun to change. Now, when you ask lay people, they will often tell you about some of the positive things that microbes do. If you ask them where microbes are, many people will know, oh, they're everywhere. That's a big change. That's a departure from the past. So where did this change come from? Well, one of the facts that has stunned people is that the vast number of cells in our bodies are microbial. So it's thought that there are about 10 times as many microbes as there are human cells. So it says that we are 90% microbe. Kind of gives you an identity crisis, huh? We know that fewer than 1% of the genes in our bodies are actually ours. The other 99% all come from the microbes, not one species, but distributed across all the hundreds, possibly thousands of species associated with us. So not only are the number of cells, of course the microbes have smaller cells, but they do a lot with those small cells, and they have a tremendous variety of those cells that we don't have. Not only do we have more of them, way more than we have of us, but they have a genetic diversity that is way greater, probably 100-fold greater than the genetic diversity in a human. So we have all of these genes carried in our bodies within the microbes. What are they doing there? So we've known for a while that microbes produce vitamins. So for example, we've known that vitamin K is a vitamin that most people don't have to take. We don't become vitamin K deficient. And the reason is that there are bacteria in our guts that produce vitamin K. And sometimes when people have been on long-term antibiotic therapy, they lose the bacteria that produce vitamin K, or the populations are significantly reduced, and they have to start taking supplements or they would bleed to death because vitamin K is important for blood clotting. So that's one example of where we kind of had a sense that the microbes might do something important for us. Then a few years ago, it was found that the microbes are critical for the development of our immune system. And this was demonstrated first in mice, and it was found that if you took baby mice, newborn mice that didn't have any microbes in them, their immune system would never develop. And if you added back one of the dominant microbes in the adult system, then they would have a normal immune system. So that started to suggest that we have a much more intimate relationship with these organisms than we anticipated. They're not just lurking in us waiting to cause disease, but in fact, our evolution has been shaped by them, and we can no longer live on our own. We depend upon them in order to develop normally to our potential. More recently, there's a lot of evidence suggesting that the microbes can prevent disease and protect us from infections. And today, we're wondering whether we can even use them in cures, and that work is just beginning. We know that in some tissues, the microbes form a barrier to invasion by pathogens. So just by a simple competition process, if the beneficial microbes are there, the pathogens won't get a foothold and infect the body. That's probably the simplest mechanism by which microbes protect us. There are many, many others. And so one of the things that we know is that we don't know much, and that will be a theme tonight. There are many, many mechanisms by which microbes make us healthier, and we understand only a few of them and only a few of the organisms that practice these mechanisms. So there's much more to find out. But there's one thing that many people have missed, and that is that even close to 100 years ago, we knew that microorganisms were absolutely fantastic gifts from nature. And that was because of their ability to produce a group of drugs that we now depend on as a society. And that is, what's going on? That is the production of antibiotics. Is there anyone here who has never, ever taken an antibiotic? Not a person. So even those of us who try to avoid taking antibiotics have taken them at some point in our lives. And it turns out that antibiotics not only treat bacterial infections, they kill the bacteria or prevent them from growing, but most antibiotics are produced by bacteria. So the soil, bacteria in the soil, have been the source of most of our antibiotics. About 3/4 of the antibiotics in current use are derived from soil bacteria. So it's a very fascinating equilibrium. We, of course, give the bacteria a lot of blame for the diseases, but in fact, it is also bacteria that make us well because they provide these fantastic drugs. And I would argue that at least in the 20th century, these are the most important drugs discovered, and perhaps in human history generally. So there's a particularly heartwarming story that I think is worth sharing about the discovery of the strains of fungi that produce penicillin. So you've probably heard of Alexander Fleming, who in 1928 discovered that there was this mold. In this case, we're not talking about a bacterium, we're talking about a mold. There are a few molds that produce antibiotics, but don't give them too much credit. It's the bacteria that do most of the work. So there's this mold called penicillium that produces an inhibitor of bacterial growth. And so here we have the penicillium mold, and what has been done here is the bacteria have been streaked on the plate, and you can see that there's an inhibition here. The bacteria won't grow close to the mold because the mold is secreting a chemical that then diffuses through the Petri dish. And so this was the first discovery by Alexander Fleming. But between 1928 and the 1940s, there were no antibiotics on the market. In fact, when Fleming was approached by an earl in England whose son was very, very ill with pneumonia, the earl said, "You have to help me save my son." Fleming said, "There's no way I have enough penicillin, this chemical that is diffusing here, to make a difference in your son's health." The earl begged him and promised him all sorts of things which Fleming probably didn't need, but it stimulated him to think of a way to do this. And so what he did was he treated the boy with the penicillin that he had, and then he collected his urine, and he isolated the penicillin from the urine, and then fed it back to the boy, and kept doing this until eventually he recovered. And so that was the first example we know of successful treatment with penicillin of a bacterial infection. So the problem for all those years between 1928 and World War II was that we had no way to make enough of the penicillin to actually become a useful drug. And so that was what Fleming struggled with, and many scientists after him. And a quite incredible scientist that I had the pleasure of knowing at the end of his life was Ken Raper. He worked for the United States Department of Agriculture at the time before he moved to the University of Wisconsin. And he worked on these very obscure fungi that live in soil. And it was completely basic, fundamental research. He had no interest in solving real world problems. But during the war, World War II, he began to recognize that penicillin could really make a difference in the outcome of the war. And so he decided to turn his interests for a few years toward the problem of finding penicillium strains that produced enough penicillin to go into commercial production. And so at the time, he worked for USDA in Peoria, Illinois. And so he put out the call across the town of Peoria, asking people to bring their moldy fruits and vegetables and bread that have that beautiful, you may not think it's beautiful, I do, but that beautiful blue-green sheen on them, that fuzzy look that they often will get when they've been sitting in the back of your refrigerator for too long. And so his technician was part of this movement, and she went out and started looking for moldy fruits and vegetables. And her name was Mary Hunt. And this is a painting. I have a copy of this in my office at the White House. If you want to see it, stop by. And you can see Mary holding a cantaloupe here. And that bluish-green growth on the cantaloupe is a penicillium mold. Mary's strain that came from that cantaloupe, which she isolated back in the lab, to this day is the greatest producer of penicillin ever discovered. So 60, almost 70 years later, we still have never found a strain that can compete with that one. It was one of those quirks of chance that changed the outcome of a war. And really, you could imagine if there was one little difference in this story, we probably would not have had enough penicillin to treat troops during the war. Well, Mary has now become known to bacteriologists as Moldy Mary. Most people don't know her last name, so I like to mention it just so that we know that she wasn't just moldy, but she was Mary Hunt. And her name was apt because she went on the hunt for penicillium strains and found the winner that has never been duplicated. As a result, and this is an ad from Life magazine that says, thanks to penicillin, he will come home, referring to troops in Europe. As a result of this strain of penicillin, which immediately went into production, so the drug companies knew right away that this was going to change the course of history. They immediately started producing large amounts, and in the early 1940s, there was enough penicillin to ship to the troops in Europe. The result was that World War II was the first war that we've ever been involved in which more people died directly of bombs and bullets and wounds than of infectious disease. And that is a pretty remarkable change. If you look back over the accounts of, for example, the Civil War, the disease, the infections of wounds were just horrific. And that's where, of course, all sorts of limbs were cut off because they had to get rid of the gangrene, which is a bacterial infection. And it was the major cause of loss of life throughout history. In fact, the Civil War was particularly horrible that way. And so since then, infectious disease has not had the hold on the human race that it did before. In fact, people can go to war and die, I'm not saying this in a positive way, but die of bullets and bombs and not necessarily think that if they get a wound, they would end up dying of an infection. So this was a pretty dramatic change in history. And after the war, the companies continued to produce antibiotics for the civil population, and they became part of general medical practice. And then people started discovering more and more antibiotics produced by soil bacteria. And so it was discovered that there were literally thousands of strains of bacteria in the soil that produced different antibiotics. And those strains were isolated. Many, many drug companies jumped in the fray. And they began to make the cultures, grow the cultures that produced these antibiotics. They purified the antibiotics from them, and then those would be the commercial products if they were safe and demonstrated to be effective. And so for decades, we were producing fantastic antibiotics, discovering lots of new ones on a very, very steady basis, until about the 1980s, when a few people got the idea that infectious disease was done. We really knew what to do about it. And a lot of the drug companies pulled out of antibiotic discovery. And that was the reason they gave, that we've solved this problem. We have all these antibiotics. We can treat infectious disease. But in fact, probably the real reason was that antibiotics just aren't very lucrative because people take them, they're sort of, their demise is their efficacy. People take them for 10 days, 5 days in some cases, sometimes even one dose, and they're done. So they only have to buy a small number of pills. Something like Lipitor, for example, or diabetes drugs. People have to take them for their entire lives. And so the drug companies make much, much more on those products. And so one by one, the antibiotic discovery companies pulled out, started moving toward other drugs, until as of a few years ago, we had only 5 major drug companies that did antibiotic discovery. When I moved from Wisconsin to Yale, I was all excited because Pfizer drug company was just up the road in Groton, Connecticut. And I thought, this is great. I work on antibiotics. I would have this fantastic collaborator. And between the time I accepted the position at Yale and I actually moved to Yale, Pfizer dropped out of the antibiotic business. And so that was a very personal example of what was happening. They're just dropping like flies. And so now we only have 4, and who knows, by tomorrow morning we might have fewer because we never know when one is going to drop out. We only have 4 major pharmaceutical companies doing antibiotic discovery. Fortunately, some of the small biotech companies have also taken an interest in this. And it may be that they will save the day because the profits are, of course, much smaller for antibiotics than with these other drugs. But to a new biotech company, a $50 million drug may sound pretty good. It may not be worth it to a gigantic pharma, but it could be to those small biotech companies. So we hope that that will continue. What has happened at the same time, while the antibiotic discovery process has plummeted, and we went from a number of antibiotics being released every year to going sometimes years without a new type of antibiotic being discovered, and maybe one being released per year, and most of those were just variations on the antibiotics we already knew. So this was a really radical change. At the same time, when we don't have new antibiotics coming along, there was also a concurrent rise in resistance, which meant that the bacterial populations that we'd been treating successfully with antibiotics for decades started to evolve resistance to those antibiotics. That meant that the bacterial mutants or genetic variants that could grow in the presence of the antibiotic would take over the population, treat with the antibiotic, and all the sensitive ones disappear, and the only ones that will remain are resistant. And so these resistant strains, sometimes called superbugs, sometimes called multiply drug-resistant strains, started to spread throughout human environments and through human bodies. And we now have pretty dramatically resistant strains of just about every major pathogen and presenting a problem, with one I think overriding all of the others being the TB bacterium that causes tuberculosis, where we have multiply drug-resistant strains that spread rapidly in dense communities, particularly cities, and we don't, in some cases, have any drugs that will treat them. Sometimes we have one drug remaining that they're still sensitive to. So this is getting to the point of being dire. We are on the verge of having epidemics with bacteria that are not treatable with antibiotics because we've selected for the ones that are resistant, and we've stopped discovering new antibiotics, and so we don't have anything coming down the pike. And this is a crisis. There's no question about it. It's been identified by the World Health Organization and FAO, and of course the United States government, President Obama, many, many organizations and governments as one of the top priorities in health security across the world. And there's a lot of collaboration among countries now to try to figure out what we can do to slow the evolution of the resistance and then perhaps encourage discovery of new antibiotics. So we're under threat now of going back to the pre-World War II era, where most people actually died of the bacterial infections rather than recovering from them, which of course was not the case during most of our lifetimes. And so it's kind of a scary time for anyone who remembers what bacterial infections can do, or anyone who's had an untreatable one, or had a loved one who has had an untreatable infection like MRSA, very common one in hospitals these days. It's a pretty frightening event. My own mother died of antibiotic-resistant bacteria, and that was one of the really sad ironies of my life, that I have spent most of my career trying to discover antibiotics and understand the nature of resistance to antibiotics in bacteria, and I couldn't do anything for my own mother when she was infected by these multiply drug-resistant strains. So it's a pretty serious problem, and we need to be looking at it very, very carefully, which fortunately this administration is doing, but I don't know that that will persist or that we will have the cooperation from other countries to really make a difference in the spread of these strains. So to move on to happier topics, the 20th century was really a golden age of microbiology. And our appreciation for microbes and dealing with them, dealing with the pathogens, really grew by leaps and bounds. So first of all, we began to understand what actually caused infectious disease. Before this, most things were known to be contagious, but we didn't actually know exactly what that meant. And then the understanding of what the basis of infectious disease was and how infectious disease spread, with these microorganisms being the causal agents, began to be elucidated across the organisms. The understanding of disease also led to an understanding of the immune system. And so the biggest barrier we have to infections by pathogens, and our best protection, is our own immune system. And in fact, the antibiotics couldn't do their job without the help of our immune system. It's very difficult for them to knock the populations of bacteria low enough that the disease would be cured. But what typically happens when we take antibiotics is we drop the populations by maybe 1 million-fold, and then our immune system moves in and takes care of the rest. But it can't really deal with the massive onslaught of a typical bacterial infection, and that's how the immune system and antibiotics work together. So this was a pretty amazing age to begin to figure out how to use vaccines, how to capitalize on the immune system with vaccines, what causes disease, and then, of course, antibiotics. But it turns out that we were missing a lot. And now we can go back and we can ask, in the late 20th century, what didn't we know about microbes, and how did things change, and why? Well, in the latter half of the 20th century, it became clear that we knew for all of that time and the previous 100 years that most of our knowledge of microbes came from those that would grow on a petri dish. Probably everyone's seen an image like this. The petri dish was invented in 1878 by Robert Koch, a doctor in Germany, and it became the standard. It just swept through the microbiological field throughout the world. And so we would culture the organisms, and we would get them into what we call pure culture, where there was just one species there, one strain there, and then we could study them in isolation. And there was a purity, a reductionism that was very appealing to the human mind for the entire 20th century. But what we discovered, what became clear from work done from about the 1950s on, was that there are actually a lot more bacteria in any environment you look at than we could culture on our petri dishes. But scientists didn't want to hear about that. So during that time, between the 50s and about 2000, scientists wanted to be reductionist. It was an era in biology where we wanted to isolate single factors and look at cause and effect. It was an exquisite era of biology. I think one of the most beautiful times of any science, because we did learn mechanisms and how cells work and how DNA encodes the functions that make cells function and the nature of genes. So all of that was great, but what it didn't account for was the big, messy stuff that is really the description of the real world. And so the scientists that were showing that, in fact, most microorganisms out in the world don't grow on Petri dishes were kind of ostracized, and they were an annoyance. And the rest of the microbiological field just said, we don't really want to think about that. We want to think about E. coli and individual genes and pure culture, and you go worry about these other microbes. So what changed that? And I think that there was one particularly dramatic finding. And so it came with the understanding of ulcers. So this is a peptic ulcer from the stomach. Peptic ulcers were commonly, when I was a kid, and probably many of you remember this, they were commonly ascribed to eating hot food or stress. Those were two of the big reasons that people supposedly got ulcers. But what became abundantly clear was that, in fact, ulcers are caused by bacteria. And Helicobacter pylori, a very lovely bacterium, I think, not if you have ulcers, obviously, but it's a very pretty bacterium, Helicobacter, because it actually forms helices. Turns out to be the essentially sole cause of ulcers, and you can now treat them with antibiotics. And there was a lot of human suffering due to ulcers because of the ignorance that we had about what caused them. And for decades, there was a resistance to thinking about any other cause besides the ones that were ascribed. But there was this pair of scientists in Australia who insisted that they had this very, very strong connection between the presence of Helicobacter and the presence of ulcers. They could not convince the medical establishment that these bacteria were causal. And so one of them actually decided that he had to do the experiment that would convince people. So he drank a very large culture of Helicobacter, and he developed the kind of irritation that precedes ulcers, and very, very rapidly. And then he took antibiotics, and he found that the ulcer condition went away. That was finally the kind of data that convinced the scientific world, and eventually he got the Nobel Prize. It has a very happy ending. But for years, people didn't want to hear that. And the reason was that Helicobacter is not culturable, or it wasn't at that time. So for decades, and when I learned microbiology, this was certainly true, people thought the stomach was sterile. They thought, how could anything survive in that highly acidic environment? Well, it turns out the stomach's not sterile. There are bacteria that are very hardy that do just fine in acidic environments, either in the human stomach or out in the real world. And in fact, Helicobacter is one of them that can survive quite well under those acidic conditions. So just because it wasn't culturable, there was no belief that it was there. And so that really was a barrier. And I think that the medical establishment was caught a little bit unawares and surprised by the fact that there was an unculturable organism that had this significant biological effect that caused a disease. So now they were no longer pushing aside these unculturable millions that people talked about out in the hot springs of Yellowstone or in soil. No, this was now an uncultured organism that was in the human body and causing disease, and therefore the unculturable world had to be taken seriously. Now, I don't have proof that that was the turning point, but I think it was a very, very dramatic moment in microbiology when people said, whoa, these things can actually matter. They're not just a biological anomaly. So in the late 20th century, a couple of scientists pioneered methods that allowed the detection of bacteria by methods that were not based on culturing. And so they began to use molecular methods that were based on DNA and RNA, the molecular molecules of life that are called nucleic acids. And these techniques that they used could be used on DNA or RNA that was extracted directly from a community. So the bacteria didn't have to be cultured and then extracted. They were just taken from their natural environment, broken open, and the DNA or RNA was taken out. And they began to show, using these methods, that in fact we were culturing even less of the world around us than we thought. That in fact it wasn't 1%, which originally people had said. No, it was probably 1 in 1,000 of the microbes in a given environment are culturable. And that's probably the extreme in oceans and perhaps in soil. In the human gut, it's much higher than that. We can culture much more effectively. But in some environments, we don't have a clue what's there. So all of what we learned in microbiology, which is substantial, this is an incredibly productive field, for over 100 years, was based on 0.1% of the species out there that are microbial. So we have a lot more to learn. And people began to ask, all right, so if culturing only captures 1 in 1,000 organisms, what is it that we haven't found yet? What's the other 99% or 99.9% out there? Who are they? And how do we learn about them? So the molecular methods that people were using to discover these organisms just provided markers for who these organisms were and what they were related to. It didn't tell us very much about their physiology or their capabilities to live in the world. They did, however, give us a beautiful new description of life. And so the tree of life, many of us probably in this room learned the 5 kingdom model. Did anyone hear that in high school? Yeah, most of us did. And it's still taught today, even though it's completely wrong. And people are resistant to change. And what happened when we started using these molecular techniques that went after the nucleic acids was that we discovered that the world is divided into 3 domains. And this is a division much higher than the kingdoms. And these were bacteria, archaea, and eukaryotes. And the prokaryotes, or single-celled organisms that do not have a nuclei, were the bacteria and the archaea. But the big surprise was that the archaea were as different from the bacteria, even though under the microscope they kind of look the same. They're little tiny cells, and they don't have a nucleus. But it turns out they are as unrelated from an evolutionary standpoint as the bacteria are from the eukaryotes. So it says that bacteria are as different from their archaeal buddies as they are from us. Isn't that amazing? And so there are these organisms that have been in nature for probably about 3 billion years, or at least their ancestors arrived around then. And they have been evolving and diversifying for all of these millennia, and that meant that there was plenty of time for the bacteria and the archaea to branch away from each other. Interestingly, the other thing that this tree of life demonstrated that is very well supported from evolutionary theory and from mathematical calculations was that most of life, even apart from the fact that most of evolutionary life was bacterial and archaeal, most of life was microbial. And what you see up here in the circle are the eukaryotic organisms that are multicellular. And those are the animals, the plants, and the fungi, like us. Many, many cells. You can see them with the naked eye. The rest of the entire tree of life is made up of microbes. So whether they are prokaryotic, meaning they have no nucleus, or they are eukaryotic, meaning they do have a nucleus, and the one that I fell in love with in 7th grade, I think, is here. It's a eukaryotic ciliated organism. Those are all one-celled organisms. So you look at the vast majority of life, and for an awfully long time, life was evolving even after eukaryotes came along, which are the most recent addition to the world. even after that, these organisms were evolving for an awfully long time before the multicellular organisms arrived. So we represent this tiny speck in the diversity of life, and it's somewhat daunting, but also, I think, places us in our context much better to recognize that this fantastic diversity of microorganisms exists in the world, and we're just a tiny portion of life on Earth. So at this point, scientists knew that we could detect these organisms using molecular techniques that didn't depend on culturing, but we would just find these markers for them that gave us a phylogenetic or evolutionary tree, like the tree of life. They didn't give us the rest of these organisms' genes and functions that would tell us what they're doing on Earth, not just what they're related to, but what do they actually do to make a living on Earth. And that started the concept of the microbiome, which means a collection of organisms in a particular habitat. So the soil microbiome is the collection of bacteria or microorganisms that live in the soil. The human microbiome would be all the organisms associated with the human body. And then the word that I came up with was to study the genomes of these organisms. I called it what we now know as metagenomics. And that means, from the Greek, meta is at a higher level, a transcendent level. And so what that meant was we were looking at a transcendent view of genomes, a collection of genomes, many genomes together. And so what we would do for these methods is to extract, just like we did for the previous methods that told us the tree of life, we'd extract DNA from the organisms right in their natural environment. So we would take a sample of soil or a sample from the human skin, not grow those organisms in petri dishes, but just extract their DNA so that we would have their DNA in proportion to their representation in that community. And then we could study the DNA and figure out what it does. And so some people started sequencing the DNA, so figuring out the order of the bases. And that told us an awful lot by comparing with other DNA sequences. We could figure out what kinds of functions were predominant. But it was dependent upon the ability to recognize genes as having a particular function, which means you had to have discovered one of those genes earlier and known its function. So there's a circularity there that we would never be able to discover new genes if we didn't find a different way and not just by comparing sequences. And it turned out that in some of the metagenomic gene banks we were looking at, as many as 80% of the genes didn't match known genes because these organisms were so new. So we know most of the genes in, for example, E. coli, what those genes do. But in fact, the wild organisms don't match the E. coli genes. Half or sometimes 3/4 of their genes are new. We don't know what they do. And so that is the cost of having studied one organism or a small number of organisms in depth for 100 years. And now we need to branch out and begin to study those other genes. And so my work led to extracting the DNA, but then instead of just figuring out the sequence, we put those DNA fragments in culturable bacteria, like E. coli or bacillus, something that grows readily in culture, and we would ask what new characteristics did those culturable bacteria acquire from having these new fragments of DNA. We might not know where the fragment of DNA came from because we're working with a gemisch. That's what the meta means. We're working with lots of genomes together. We cut them up, put them in different organisms, and look for new functions. And from this work, we found new antibiotics that had not been found in cultured organisms. And we found many, many antibiotic resistance genes that are not in the cultured organisms. And we think that that might be significant because we may be able to anticipate where the antibiotic resistance genes come from and when they're going to move into the cultured bacteria that, or the pathogenic bacteria that we see in the clinic setting. So this gives us a new window on gene function, because we've seen these genes that can make an organism produce an antibiotic or make an organism resistant to a known antibiotic. And this begins to broaden our view of the genes and the organisms that are out there. So metagenomics has led to a lot of discovery that you're probably hearing about in the news quite a lot. And it's been quite a fascination recently of the public, because we have discovered that there are these pools of genomes that we've never seen before that come from the uncultured organisms. And we now have ways of characterizing those genes without culturing the organisms. So some of the questions that people have asked with metagenomics and studying the microbiomes, the collection of microorganisms, are things like, okay, if we have 100 trillion bacteria, 10 times more cells of bacterial type than our own, what in the world are they doing in our body? Presumably, we evolved with these organisms in some way that developed a codependence. Otherwise, we probably would have kicked them out by now. But the experiments are hard. So I'd just like to show you, I think there's some really elegant experiments that get at the roles of these microbiomes, or the organisms that live in us and on us. So an interesting example is the human colon. So we know that any individual person has about 500 species in them. And across all humans, there are probably more than 1,000 species. We all have a different composition of microbiomes, so a different set of species. There's some overlap, and there's some thought that there's a core that everybody has, and then everything else varies. But we don't really know yet how that works. And so there's been a lot of work to try to characterize what are the species and what are the functions, using metagenomics, that those organisms carry that make a typical human gut, and what makes a unique human gut. So what makes your gut different from your gut. And so that work has led to a lot of descriptive analysis. So describing a lot of guts and saying, oh, there are these bacteria here and these there, and pregnant women have this composition of gut, and when they stop being pregnant, then their gut goes back to before, and all these different analyses. But it really didn't tell us what those organisms were doing. And one of the most elegant approaches, in my opinion, is one that was pioneered by Jeff Gordon, a professor at Washington University in St. Louis. And this has been used to characterize the microbial contribution to a number of different diseases. And the experimental approach is this. He would take the human microbiome, so a sample of whatever habitat in the human body he was examining, and he would transfer those bacteria, no culturing, just take them en masse, and transfer them to germ-free mice. So these are mice that are born by cesarean section to mothers that have no bacteria in them. So we call them germ-free. And he would then look at, if you take these organisms, and this is not a Petri plate that's intended to be under the microscope, so you take this whole mixture of different microorganisms, you transfer them to the sterile mouse, and then you look at what Jeff Gordon would call the humanized mouse. So what changes in this mouse when you introduce the human microbiome? And one of the most dramatic results was when he took the microbiomes from obese people and thin people, obese and lean. He transferred them to germ-free mice, and he got results that looked like this. So this poor little mouse has the microbiome of the people who were obese. This mouse got the lean microbiome, and there's been no change in weight. And there have been all sorts of experiments since then to try to manipulate the microbiome, to try to reverse the effect of this transfer, to look at the effect of diet on the nature of the microbiome. But this was really kind of a seminal finding and suggested a very creative way of looking at function. We can't do those experiments on people. You know, we can't knock out the bacteria, make germ-free people. That's not ethical. And so Jeff was able to use the power of the human microbiome, but transferred to an experimental system that was otherwise clean to study cause and effect. I want to tell one more story about how another disease was studied, and that was autism. And so a group at Caltech, Sarkis Mazmanian's group, they have a way of making the pups, young mice, autistic or have characteristics in common with autistic children or people by treating the mother while she's pregnant with some sort of stressor. So it turns out that you can induce a pretty high frequency of autism by stressing the mom when she's pregnant. So then the pups would begin to show autistic behaviors, which are characterized, one, in antisocial behavior. They wouldn't make eye contact with their buddies. And the other was obsessive behavior, where they would bury a marble and then dig it up and then bury it again and dig it up over and over and over. And these have parallels in human behavior. So it's seen as a proxy for studying autism. And what Sarkis found was that when he looked at the guts of the pups that had autism symptoms, he found that this is the lining of the gut here in diagram. He found these holes in the gut wall that seemed to be allowing for the passage of chemicals, maybe other things, but certainly chemicals, into the rest of the body. So normally these chemicals would be contained in the gut because you have an unbreached barrier there. All of a sudden, when he looked at these pups, they began to have little breaches, and he thinks that what happens is that neurotoxins that are produced in the gut, either by the microbes or the humans, get out into the system and affect brain activity. He found that there was one particular bacterium that was consistently missing, and that was called Bacteroides fragilis. And so he took that and added it back to the leaky gut mice, the mice that had autism symptoms and the leaky gaps in their guts. And he found that these leaky spots closed up, the junctions between the cells in the gut lining would close and seal, and some of the autism symptoms went away at the same time. He found that although the mice didn't become any more social, they still wouldn't make eye contact with their friends, they wouldn't do their obsessive behavior of burying the marble and digging it up again. So there's clearly more to this. The entire suite of autism symptoms are not explained by this result. But it's a pretty dramatic result, especially given that children who have autism very often have what's called a leaky gut syndrome. And so there's a tremendous parallel of a bacterial difference that seems to typify the autistic gut, a physiological difference of being leaky, and of course the behaviors of autism. And so it is going to be really interesting to see if this result, where he restored a normal condition using Bacteroides, can be replicated in humans. Lots more to do before that happens. So I hope you've seen a little bit about what microorganisms do, where our historical view came from, and hopefully gave you the evolving view so that you now love your microbiome and love your bacteria and honor them as they should be honored. We still have lots to answer of how we figure out what they do and who's there, and figure out new ways to study these microbes. And the future, I think, is pretty exciting. So one of the things that we've seen repeatedly across this field is that the same questions come up over and over, whether we're looking at the microbiome of a human or the microbiome in the Gulf of Mexico, for example. And those sound like really different habitats. But let me give you one example. So you may have read, this is kind of gross, I'm sorry for right after dinner, but you may have read about fecal transplants. So these are used with people who have this disease known as C. diff, which is a sometimes fatal, extremely debilitating disease that usually occurs after long-term antibiotic or very extreme antibiotic application. Results in 250,000 hospitalizations every year, 14,000 deaths, and a lot more suffering than that would indicate. 30% of the patients don't respond to antibiotics. So this Clostridium difficile that causes it is either not responsive to antibiotics or is somehow protected from the antibiotics by the environment. And a good portion of the patients don't recover. And some of them get very, very close to death. Well, many of them die. But they end up essentially wasting away. And so one of the scientists who looked at these people found that their microbiome was very, very simple. They had fewer organisms that a typical microbiome would have. And so they took a chance, and this was a microbiologist and a physician got together, and they transferred the microbiome from a husband to a wife, and they've now done it with many different people who live in the same house, which turns out to be the people who have the most similar microbiomes. And the woman, the first one treated, recovered in 24 hours. She got up and walked away out of the hospital, and she was fine. She was literally on death's door. This has now been tested over and over, and 95% of the patients will be cured in 24 to 48 hours after suffering from this disease, usually for weeks, months, and even years. And they, at the beginning, only used it on the most extreme cases who were really going to die anyway because they were unresponsive, because it seemed like a really big risk, taking fecal matter from one person, putting it into another. It was unlike anything that had ever been done. Now it's used a little bit more widely on people who are not just about to die, but are just suffering. So that's an example where we take a whole gamish of microbes, transfer them, and we see this effect. We can't say what the organisms are that are important to that. We just say the transfer has this amazing effect. But if you go back in history, there are parallels with this. And if you go back into Roman times, we know from writings of that time and then traditional agriculture ever since that you can plant plants in a field, certain plants, and it's always the legumes, and they won't thrive very well. They'll often turn yellow. But if you take a scoop of soil from a field where they'd been doing well and sprinkle it in the new field, they will turn green. And so this has been known for decades, never for centuries. And it was clear, and it took a long time to figure out what was going on, but it was clear that there was sort of a contagious factor that you could transfer. And it's just like the fecal transplant. You take a little bit of the microbes, put them in this other environment, and you can fix a problem. The other parallel was with what we call suppressive soils. Some soils just naturally suppress particular diseases. The plants just don't develop that disease, even if the pathogen is there. And again, you can take a scoop of soil from a suppressive soil and put it into a field that is very susceptible. And after that soil has been spread and a couple years have gone by, you can make that new soil suppressive as well. So once again, we have examples in these very, very different environments, the human gut and agricultural settings, that are behaving very similarly. And so that has led us to ask a number of questions across all of microbiology and all microbiomes that lead to some very practical changes that we want to make in microbiomes. So the first is, how do we discourage a community from being invaded? So that you might want to do if you're trying to prevent the invasion by a pathogen. And the same thing is true, the same question occurs whether you're dealing with the Gulf of Mexico or the human gut. But we don't know the mechanism. We don't know how communities resist that invasion. We also don't know how communities that do change recover. And this is actually a real issue and becomes a very interesting one financially and in all sorts of other ways, legal ways. So we know that when you treat people with antibiotics, their microbiome will go kind of nuts for a while. The species will change on almost a daily basis. The composition doesn't look anything like it was before, and it's changing radically. But by about a year later, the microbiome will have settled down and developed what we call a new norm. We don't really know if it's normal, but a new type of microbiome that's pretty stable then. I went to a meeting last year, and last fall, and I heard this talked about in a meeting that was about animal microbe interactions. And the question was, what is a healthy microbiome? Because we know that you treat with antibiotics and you get a change, but we don't know if that change is good or bad or doesn't matter. And so we really need to understand how the community bounces back if it does. How does it acquire this new norm, if that's what it does? And is this better or worse? Maybe it's better, maybe it's not. We assume because the antibiotics have changed it from some other normal state that it's not as good. A week later, I went to a meeting on the Gulf of Mexico and the recovery after the BP oil spill. And I heard in almost exactly the same language, how do we know what a healthy microbiome is? We know that in the Gulf, after the oil spill, the microbiome went wild. It was very unstable. New species seemed to appear, old species disappeared. The composition was new. But after about a year or two, it settled down to what we call a new norm. And the answer, or the question that the scientists were asking is, should we charge BP reparations for reparations because they've ruined the microbiome? Well, we don't have a clue, because we don't know what a healthy microbiome is. We don't know if the new microbiome is healthier, less healthy, or the same. So it's really quite striking that these two very different systems, the human gut and the Gulf of Mexico, had these very similar patterns, and the questions were the same. And then the third type of question we'd like to ask is, how do you discourage recovery? So what if you change the microbiome in a good way, so the person or the Gulf or the soil is healthier? How do you make it not go back to its original state? And once again, we don't have a clue. And so one obvious example that you may have heard about is probiotics. We know that you can get effects from taking probiotics. Those are bacteria in a pill, if you take it every day. But as soon as you stop taking those probiotics, the populations of bacteria disappear from your gut. Why is that? How come they don't establish? We don't have a clue. So again, asking why these microbiomes recover to their original state and kick out an invader is a critical question. So you may have seen, there was an article in the New York Times last week about scientists urging the government to launch a national initiative that they call the Unified Microbiome Initiative. And that was based on two papers, one in Science magazine, one in Nature, that urged us as a government to develop these integrated approaches that would cross-cut and begin to ask some of these questions instead of in isolation in the human microbiome silo and the aquatic silo. We would begin to ask those questions across different habitats and focus on the question rather than where we're asking it. And these were very, very well thought out, very interesting papers. And they came after a series of meetings that the White House had held, and we had done a number of other things to try to gather thoughts from the scientific community. And this was the theme, this was the analysis that resulted. So we don't know yet what we're going to do, but as I was quoted as saying in the New York Times article, we have nothing to announce today. And I'm hoping that one of these days I'll be able to say we have something to announce, but I don't. And the White House is taking these recommendations of the scientists under advisement and trying to develop something that will meet the needs of the scientific community as well as the federal agencies and answer some of these really big questions. I just want to point out that in forgetting our microbes throughout history, by ignoring them, we have suffered. This is an example of Biosphere 2, which was the experiment set up in Arizona, where a simulation of the Earth was built, and they did all the calculations they thought they needed. They put people into this simulated ecosystem that had the ability to produce food and deal with waste. Within 6 months, people could no longer live there because the oxygen concentration had gone down so precipitously. And the answer was that the microbes in the soil were using up the oxygen, but nobody bothered to take them into account when doing the calculations. And there are numerous examples like this over the history of science and microbiology as well as the rest of biology. We forget about the microbes because we can't see them. And Biosphere 2 was, as a result, a failure. But we can't afford a failure on the big biosphere. We really need to make sure that we get it right. And hopefully, as humans begin to answer some of these key questions, we'll begin to figure out what the microbes are doing that keep our planet sustainable and healthy. And with that, I'd like to thank you for your very long attention and stop there.
So we have time for questions. There is a procedure for the questions. If you'll keep your hand up, one of the runners with microphones will come to you. When you get the microphone, please stand up, tell us your name, ask a question. No speeches until after and the social hour, and we'll proceed that way. So you can.
How do you, sorry. How do
name
you
and if you're a member.
identify an actual microbial genome or the area in which you're going to look for the microbes? And you say you can't culture the individual ones, but you're looking at the DNA from them. So how do you know you're getting separate organisms, or do you even, or are you just getting chunks of DNA from a bunch of different things?
What's your name?
Bruce Meary. I'm a visitor. Yeah.
That's a great question. So where do these pieces of DNA that we isolate in metagenomics come from? And how do we know that we're getting different organisms? So there are some genes that are present in bacterial genomes either one time or a few times. And so they become a marker. So every one of those genes probably comes from a different organism, and they serve as the markers for evolution that I showed the resulting tree of life. So if you have thousands of different examples of these genes that act as markers, then you can assume there are thousands of different species because they're distinct marker genes. The rest of the genes we often don't know the origin of. So we know that in the entire metagenome, or the microbiome, we have X number of genes that are involved in lipid synthesis, for example. This has linked bacteria in the gut to cardiovascular disease. So we know that the microbiome generally has a frequency of a type of gene, but we can't say which genes go with which organism unless we can culture or we play a few other tricks. And those tricks are hard. So that's one of the big questions, is how do we begin to associate the functional genes with the evolutionary phylogeny, which is the distribution of the organisms and their taxonomy, their names. And we're not there yet. So learning how to culture them is one way, actually. And that's been very successful so far with a few species.
My name is Al Ehrlich. I am a member of the Society. Did I hear you correctly when you said that these microbe-free white mice early in the talk don't develop an immune system, but if you introduce a single species of bacteria, then they do develop a normal immune system?
Yeah, so the immune system is essentially stymied if there is an absence of Bacteroides, one particular species of Bacteroides, iodomicrons is its name. And if you add that species back, then they will develop normally.
So the immune system doesn't need much to get going and do its genetic thing?
Right. Most of the genes for the immune system will be expressed without the bacteria, but without this pretty key step, it won't develop the full way.
Thank you.
I'm Dale Doucette. I'm a member. If I remember, you've found microbes at the bottom of the lowest point that we've gone on Earth. And I would like you to comment on that. And the other is they found them in the atmosphere out very far. And would you discuss that?
Almost every environment on Earth or around the Earth that we've, or in the Earth that we've looked at has viable microorganisms. And so one environment that I particularly love is the deep sea geysers where you get these steam vents from underneath the surface of the ocean. And these are really deep down, so the pressure is very high, which means the temperature can go way over boiling. And you see, there are beautiful pictures of them essentially boiling off. And there are bacteria in there. There are microbes living in those environments. In the Yellowstone Park, if you go to the highly acidic, very, very hostile environment for most organisms, there are environments that are pH 1 or pH 0, I mean, really, really acidic, and there are bacteria living there. There are environments there that are superheated to 113 degrees centigrade. That's way over boiling. There are bacteria living there, bacteria and archaea. So far as I know, any of these extreme environments has been shown to have life, and that's been done with gold mines in South Africa, where they've drilled very deep in the earth, miles down. They find microorganisms, and above the earth as well. And we know that bacteria exist in pretty reasonable concentrations through the atmosphere. And then when you get to the stratosphere, I think it's a little bit more ambiguous, but there are probably some up there. There's a lot of interest right now in the bacteria that live in clouds, because it turns out that there are bacteria that cause the nucleation of ice, which means you take water which is colder than the freezing point, and you add ice and the water will freeze. Well, it turns out there are bacteria that are the best known ice nuclei. You can take this supercooled water, add these bacteria, and even just a little bit below the freezing point, they will cause the ice to form. So the question has been for a long time, why would bacteria have the ability to cause ice to form in supercooled water? What's the advantage there? And a hypothesis that came up about 20 years ago and is now finally beginning to be tested is that the place that is really important for ice nuclei to exist is in the clouds, because as ice nuclei form there, they allow the formation of raindrops, which start as ice crystals. In fact, you've probably heard of silver iodide seeding of clouds. That's what's happening. Silver iodide is the best known ice nucleus besides ice, and it stimulates the production of ice crystals, which then come down and melt and become raindrops. And so it could be that these bacteria have a role in rain induction. And that would be a really interesting, novel purpose or function of bacteria in the environment. So yes, we know that they're up and around in the atmosphere. They're at low concentrations, which makes them very hard to study. You have to fly through clouds and collect an awful lot of air before you pick up the bacteria, but they are in fact there. Did that answer both questions?
Yes.
Hi, Carl Merrill, member of the Society. I wonder if you could expand on how we're going to learn what all these genes do, given that, as you pointed out, we're basically superorganisms. Mm-hmm. And so if we try to culture some of the genes in different bacterial strains, we know of some genes, like the diphtheria toxin, that only affects mammalian enzymes. Mm-hmm. And so there must be lots and lots of those. And how do we even begin to approach something like that?
Yeah, it's a really good question. It's kind of mind-boggling. For years, scientists were and the National Institutes of Health was very supportive of this approach. They were giving scientists the means to study these unknown genes, and there are plenty of those in humans too, genes we don't have a function for, and trying to figure out experiments that would predict the gene function or discover the gene function. And that has generally not been very productive. And so the question is, how can we do better than that? Metagenomics is one way, looking for functionality in a wide variety of assays. Another way is to look at populations in an environment under various stresses and see what genes become more prevalent. And that doesn't tell us anything definitively, but it can at least pose a hypothesis of what those genes might be doing. And so, for example, genes involved in fatty acid synthesis have been associated with obese microbiomes. So that kind of makes sense. You could imagine those microbiomes are better at synthesizing lipids. So that gives us a hypothesis to test. Maybe some of those other genes that are unknown also play a role in lipid metabolism. Other than that, there are not a lot of great ways to do it. And I think that's one of the big mysteries that people are still struggling with and not quite approaching yet, because there's so many questions that we can approach at a slightly higher level of understanding that I think the next step will be digging into those genes and figuring out their functions. But right now, we're just trying to figure out what happens to the microbiome when you treat it this way or that way.
Hello? Yes. Oh, sorry. Hi, thank you. I'm Tony Lopez. This is my second talk. Thank you for a wonderful presentation. I'm looking to move into biofuels, and I'm wondering how much microbiology is being used in creating alternative renewable energies and how much success they're having now.
There's quite a lot of activity in the biofuels area. So one example is the fermentation of plant material, so the kind of generally hard to degrade plant material that's left in a field after the crop is harvested, so the stover from corn plants, for example, and finding enzymes in microbes that will digest and then release energy from those tough polymers. So that's one example. The Department of Energy has been supporting that kind of work for a long time, and there have been some really big breakthroughs in the last decade on using microorganisms to ferment plant material and produce, in some cases, ethanol, and in other cases, other kinds of, like biodiesel. Another area, I'm sorry I took the slide out because it's a pretty one, is the use of algae. These are eukaryotic algae that they grow in these gigantic glass tubes. They look sort of like test tubes, except they're really big. And they put them in the desert in Arizona and areas where there's a lot of abundant sunshine. And they let these algae grow. And they have strains that put 50% of their body mass into lipid. And it turns out that that lipid is either biodiesel itself or very closely related to diesel. Now, I think that's one of the more promising techniques because they're putting them in the desert where the sunshine is free, the energy is light and it's driving the reactions, and the rest of the chemistry is actually pretty cheap. And so I think that's going to be really interesting, promising. There are a few other examples, but bacteria are the sort of backbone of the biofuel techniques.
Hi, Frank Robert, a member. If you can forgive the point of view coming from, I guess, Harvard, Cornell, and Princeton instead of Yale, but that something like half the biosphere may be in the rocks, and that if you look at the archaea, so many of them are methanophilic and are thermophilic from our discussion of extremophiles. Have you any thoughts on the idea of the thermophilic common ancestor to the whole of the tree?
Well, I think we know that a lot of life evolved under much hotter circumstances. So yes, I think it's likely that the thermophiles are some of the earliest ones. If you look at the root of the tree, there certainly are organisms related to what we think were ancient thermophiles, thermophiles being organisms that like really hot temperatures. But I don't think we know that for sure. I mean, I think I am always a little uncomfortable with evolutionary arguments because we can't do an experiment to test them, and I'm an experimentalist at heart. So I'm probably not the best person to ask, but yes, I think there's reasonable evidence that some of the very earliest organisms were thermophiles. Does that answer the whole question? Oh, good question. I don't know. On Earth?
Hello, my name is Joe Spector. I'm a member, and I was wondering, so when I've looked at the over-the-shelf kind of probiotics that are available, I was just wondering. It seems like there's a wide range of them, and I was just wondering, is it sort of just guesswork right now, or are there a lot of that? Are there some specific ones that are well documented or researched that have specific benefits, like cardiovascular disease, or I don't know, autism or anything like that? But
Yeah, great question. We're not quite there with cardiovascular disease or autism. I hope in the next few years we're going to start seeing some advances there. There were some studies from one of the derivative companies of Dannon Yogurt that produces some probiotics that they were looking at Crohn's disease and other intestinal ailments, and they had indications that different probiotics had different effects, but even more significantly, the mixture of probiotics could change the effect of any one of them. So you mix a very helpful probiotic with another one, and you completely reverse the effect. So that kind of made people stop and say, this is a little bit more complicated than we thought. And these can be very, very closely related organisms, even within the same species. The best evidence that I've seen for positive immunological effects, some protection against ulcers and GI tract health is an organism named Lactobacillus reuteri, R-E-U-T-E-R-I, or sometimes people just call it reuteri. And you can buy it on the internet, and there's actually really good data mostly from Canada showing that it has these beneficial effects. A lot of the others, we don't really know. Maybe the companies that make them have actually tested them and have great data, but I haven't seen it. And I do know, and this is just complete anecdote, not to be spread from this room, but my lab works in part on caterpillars. And we were testing a number of things in caterpillar blood, so basically injecting them, their blood with various bacteria. And we found that one of the most commonly, the largest market share of the probiotics, one of the most commonly used probiotics kills our caterpillars. No other organism did, but this one probiotic did. And after I learned this, I was kind of nervous. Okay, I'm not a caterpillar, but it kind of gave me the creeps. And so I went back and I found a book that had actually been edited at Yale, of all places, and the physicians that wrote in it wrote about the only deaths that have been associated loosely with probiotics. And they were all in infants, and they were all with the same species that we found killed the caterpillars. So, anecdotal, not a proof of anything. What's that?
can you say which one that is?
It's called Culturelle, so probably pretty common on the shelves. Now, it's not going to get in your blood, and it's probably not going to kill you. It probably won't kill most infants. I think there have been like 9 deaths, so it's not a big threat. But that just made me a little bit nervous about probiotics. But Lactobacillus reuteri has been widely tested, very positive effects, and I think the market for it is growing. I actually gave the papers on it to Whole Foods and said, you really should stock this. You have all these others that are unproven. This one actually has data behind it, and many Whole Foods now carry it, so you can actually get it in person.
My name is Jim Griffin. I'm a member. I, as a boy, have a very clear recollection of my mother repeatedly telling me, you have to eat a peck of dirt before you die. And I, at some stage, thought that represented her farm girl growing up and a peasant ignorance of medicine. And only in the last 5 years, I'm beginning to wonder whether it was a profound folklore that permeated under the medical system and is actually a very positive view of children.
Absolutely. I don't think we have definitive evidence, but I think there's accumulating evidence that says that soil, I prefer to call it, not dirt, carries some of the bacteria that we actually need. and that maybe this is the explanation for what's called the hygiene hypothesis, that kids growing up in these very clean environments, particularly away from farms and city environments, tend to have a much higher frequency of asthma and other kinds of inflammatory diseases. And perhaps it's the consumption of soil that protects kids that grow up in rural settings from those diseases. We don't know that for sure, but there's a lot of circumstantial evidence that I find really intriguing. So I bet your mother was right. My grandmother was also right. My father tells this story that she used to take moldy bread from the back of the refrigerator and make poultices and put it on cuts and wounds. And of course, what she was probably doing was applying penicillin, had no idea. They all thought she was crazy, moldy, stinky bread, but it apparently was doing something.
Didn't the American Indians also do that?
Oh, I don't know. I never heard
I
that.
had the impression when penicillin was discovered that I heard that, the Indians in the U.S. taught the settlers to find some moldy stuff and put it on the cup.
Oh, so my Latvian grandmother and the American Indians. Thank you. That's great.
Ellen McCauley, you've moved back to the discussion of antibacterials and resistance. So as we think about the future, you've talked about discovery and identifying more antibiotics. What about, or how long do you think it would take for bacteria to no longer be antibiotic resistant? So in other words, right now it's so pervasive. But to lose that resistance, are we talking 100 years? Are we talking 1,000 years? Is it so pervasive that that's not even a way to think about it? And I know it's a tough experiment to do.
It's actually been done, though. It's a great question, because you would think that most of these changes probably aren't to the advantage of the bacteria. Only under the conditions when the antibiotic is there. Otherwise, they probably would have evolved these characteristics without the antibiotic being there. So there's probably some cost. And so the simple-minded prediction was that you have the antibiotic there and resistance will increase and increase and increase. You take the antibiotic away and it'll go back down to the original quantity at the same rate that it rose. And that turns out not to be the case. And it's been looked at entire country levels. And so Denmark and Sweden and Iceland have done some experiments of removing antibiotics from their arsenal and just stopping using them. And surprisingly, there is usually a drop. It will drop after the antibiotic is taken out of circulation. But it levels off at a much higher level than it started at, and I don't think anyone has a clue, I certainly don't, why that is. So there's a little bit more to the story than we think. One hypothesis is that we're somehow selecting for resistance with other chemicals. So one that's popular is triclosan, which is in a lot of these hand sanitizers, that maybe the resistance genes that make bacteria resistant to antibiotics also confer resistance to triclosan. So as long as we're using triclosan, we're selecting for the resistant ones. We don't have a lot of evidence to support that yet. But it's a very, very interesting question. How long will it take to go back to the original populations, or will they ever? And it sort of is related to this, what's a healthy microbiome and what's recovery? Once it levels off, does it just stay there for decades? We don't have enough data yet. We do have a few years of data and know that it drops. The one thing that has been very effective is for hospitals to cycle through antibiotics, because they use, everybody, all the doctors use particular antibiotics for particular ailments, and then they'll stop after 6 months and they'll use a different one. And in those cases, that's been actually shown to be a very, very sound strategy for slowing the rate of resistance.
I'm David Rosen, a lifetime member. What about those tabletop experiments in evolution like Lenski's 30,000 generation? Is it 30,000 now? I'm not sure what they're up to.
30 or 60, somewhere in there, yeah.
I'm not sure where they actually look at the evolution of a culture and its how it diverges. Would that help? Does that feed into the type of research you're talking about?
Lenski's work asking, so he has cultures of E. coli that go back decades, and he transfers them on a very regular basis. So some of them, as David said, are now through 30 or 50 or something like that, 1,000 generations. And so it's a great population, and he saved samples, of course, at every step of the way, and you can compare them. And so it's a great way to look at gradual evolution, and that's just under normal growth conditions. He's not applying any selection. It tells us a lot about frequencies of mutation and the way gene mutations will sort of drift in a population when there's no selection pressure. I don't know if it tells us a lot about the microbiome in terms of gene function and how populations or communities go back to their original state or resist change, but I don't know if we know enough yet. It's at a much more micro scale within a species, within a pure culture that Lenski is looking at. Yeah, right. That's another condition. So you're not getting gene exchange. That's right.
So I think with that, we'll thank our speaker. Before you go, before we adjourn to the social hour, I have a couple of things to do, the first of which is to thank our speaker.